You know, usually when we learn something in grade school, we just sort of assume it's like the bedrock truth, right? We get handed this neat little flashcard that basically says, plants breathe in carbon dioxide and breathe out oxygen.
Right, yeah. It paints this picture of a perfectly harmonious exchange. Like, we breathe in oxygen, they breathe in carbon dioxide. Just a closed, perfect loop that, requires no further thought.
Exactly. But then you step into the world of actual biochemistry, or, heaven forbid, you sit down to take the TEAS or HESI nursing entrance exams, and you realize that flashcard is a massive oversimplification.
It really is.
In fact, relying on that grade school definition is like it's a trap that will absolutely trick you up on test day.
Right.
So, okay, let's unpack this. If you look at the actual summary equation for photosynthesis, it's 6CO2 plus 6H2O plus light energy yields, C6H12O6, that's glucose, plus 6O2.
Yeah, and that single line of chemistry hides an incredibly complex two-stage energy conversion process. Photosynthesis is not simply a plant breathing,
Right.
The overarching goal of our deep dives today is to understand exactly how plants turn light into chemical energy, dispel some very persistent historical misconceptions, and, well, crucially for you future nurses out there, connect this plant biology directly to human physiology.
Welcome to the deep dive. So we are unpacking photosynthesis today, specifically tailored for those of you prepping for your exams. But honestly, if you're just a curious learner who wants to understand how the world actually works, you're in the exact right place.
Absolutely.
To really grasp what that chemical equation means, we first kind of have to look at how incredibly wrong early scientists were about plant growth. Like, they were completely in the dark.
Yeah, to appreciate the mechanics, you really have to appreciate the mystery. For centuries, the smartest minds on the planet were just staring at trees, completely misinterpreting what was happening right in front of them.
I was actually looking at Jan Baptist van Helmont's experiment from 1648.
Yeah.
So he planted a 5-pound willow tree in a large pot containing exactly 200 pounds of meticulously dried soil. And he just waters this tree constantly for 5 years.
5 whole years, yeah.
Right. And at the end of 5 years, the tree has grown massive. It weighs about 169 pounds. But when he dries and weighs the soil again, the soil only lost about 2 ounces of mass.
Right, so Van Helmont looks at this data, sees the tree gained 164 pounds, and the soil lost almost nothing. And he concludes that the tree's mass came entirely from the water he added.
Which, if you think about it, is like someone weighing themselves, realizing they gained 164 pounds over 5 years, and concluding it happened purely from drinking tap water.
Exactly.
But his data was completely sound, so why is this considered one of history's most famous right experiment, wrong conclusion stories?
What's fascinating here is that Van Helmont actually made a huge scientific leap. Prior to him, the prevailing belief among naturalists was that plants literally ate dirt.
Ate dirt, wow.
Yeah, they thought plants derived their physical bulk entirely from consuming the soil. So by meticulously weighing the soil, he definitively proved that soil is not the primary source of plant mass. We know today that soil only provides, minor minerals like nitrogen and potassium.
But he lacked the modern chemistry to see the invisible variable, right? Like, he had absolutely no idea that the mass was actually coming from atmospheric carbon dioxide being pulled from the air and fixed into solid carbon compounds.
Right, exactly.
He didn't know you could literally build a solid tree out of thin air and water.
He had no clue.
Yeah.
It actually took over a century to slowly build the theoretical framework to understand that invisible gas. Fast forward to 1771, and Joseph Priestley placed a sprig of mint in a sealed glass jar.
Oh, this is the mouse experiment, right?
Yes. He found that the plant could restore air that had been injured by a burning candle or a breathing mouse. If you put a mouse in a sealed jar, it eventually passes out. But if you include the mint plant, the mouse survives.
Providing the very first evidence that plants release a life-sustaining gas, which, we now know as oxygen. But Priestley didn't know why it was happening or what triggered it.
Right, so then Jan Ingenhousz steps in a few years later in 1779 and realizes this air-purifying magic only happened when the plant was explicitly exposed to sunlight. He also noticed that only the green parts of the plant could perform this trick.
Ah, okay.
So light was finally identified as the essential energy source driving the entire process.
And then Nicholas Theodore de Saussure comes along in 1804, runs the math, and finally quantifies the gas exchange. He proves that both carbon dioxide from the air and water from the ground are incorporated into the plant's tissue.
Exactly.
For you listening, keep that in mind. Scientific consensus rarely arrives in a single eureka moment. It's built piece by piece, often by realizing the brilliant scientist before you was, well, only half right.
Yeah, that's a great point. So now we have the basic ingredients. We know what goes in light, water, carbon dioxide, and what comes out, which is sugar and oxygen.
But that doesn't really explain how the actual factory floor works. I mean, that grade school flashcard still makes it sound like a single simple reaction. If we shrink down and go inside a leaf, what are we actually looking at?
Well, we have to deconstruct the anatomy of the process because it happens in two distinct coupled stages inside a specific organelle called the chloroplast. You have the light-dependent reactions and the light-independent reactions, which are more commonly known as the Calvin cycle.
Okay, let's walk onto that factory floor. Give me a visual of that first stage.
All right, picture the inside of the chloroplast. You'll see these microscopic membrane stacks called thylakoids. They look almost like stacks of green hollow pancakes.
Green pancakes, got it.
Right. This is where the light-dependent reactions occur. The chlorophyll and other pigments embedded in these pancake membranes absorb incoming photons of light. That solar energy excites electrons within two highly specific protein complexes, creatively named photosystem 2 and photosystem 1.
Hold on, if the light is just providing energy to excite electrons, where does the actual physical oxygen we breathe come from? Does it come from breaking down the carbon dioxide?
Actually, no. And that exact question fueled a massive debate among scientists for years. But in 1941, Samuel Rubin and Martin Kamen finally settled it.
How did they do that?
They used a heavy isotope of oxygen, oxygen-18, to tag the water molecules they fed to a plant. When the plant released oxygen gas, they traced that heavy isotope directly back to the water. At photosystem 2, the plant literally takes H2O and splits it apart into protons, electrons, and oxygen gas.
Wow. So the water splitting process is the exact undeniable source of the oxygen we breathe. Oxygen comes from water, not carbon dioxide.
Yes.
That is a massive detail for test day. But wait, what happens to the rest of the water molecule, like the protons and electrons?
Well, those excited electrons that were freed up by splitting water get passed down an assembly line called an electron transport chain. As they move, their energy is used to pump protons, the hydrogen parts of the water, into the hollow interior space of the thylakoid pancake.
Okay, so we're packing all these protons into a tiny enclosed space. That sounds like building a pressure cooker.
It's intense, yeah.
Yeah.
Think of it exactly like a hydroelectric dam. You pump water to one side of a dam, creating immense physical pressure. When you let that water flow back through a specific narrow channel, the force of the water spins a turbine to generate electricity.
Okay, that makes sense.
The plant does the exact same thing on a microscopic scale. It builds up a massive proton gradient and then lets those protons flow back out into the main body of the chloroplast through a specialized enzyme called ATP synthase.
So it literally spins a microscopic biological turbine.
It physically rotates, yes. That process is called chemiosmosis, and it generates ATP, which is the fundamental cellular energy currency. At the same time, the electrons finish their journey down the chain and are caught by a carrier molecule, turning NADP plus into NADPH.
So stage 1, the light-dependent reactions, is entirely about capturing solar energy and storing it in these temporary chemical batteries of ATP and NADPH.
Right.
But you can't build a 160-pound willow tree out of pure energy. How does the plant actually build its physical structure?
That requires stage 2, the Calvin cycle. This stage was mapped out in the 1950s by Melvin Calvin and his team at UC Berkeley using radioactive carbon-14 to track the molecules. We leave the thylakoid membrane stacks and move into the stroma, which is just the fluid surrounding them.
Okay, into the fluid.
Yeah, and the Calvin cycle doesn't directly need light. It takes those temporary batteries from stage 1, the ATP and NADPH, and uses them to physically attach or fix carbon dioxide molecules from the air into 3 carbon sugars, which eventually become glucose.
This is where the plant is literally pulling invisible gas out of the air and turning it into solid matter. And the primary enzyme that drives this carbon-fixing process is called rubisco. But from what I read, rubisco is kind of terrible at its job.
Oh, it really is. It is notoriously slow as a catalyst and highly prone to catastrophic mistakes.
So it's like a factory worker on an assembly line whose only job is to grab red boxes the carbon dioxide and put them on the belt. But this worker is incredibly sluggish, and half the time they get distracted, grab a blue box, which would be oxygen, by mistake, and jam the entire assembly line, forcing the factory to waste energy fixing the mistake.
That is a highly accurate, if slightly insulting to the plant analogy. Because Rubisco is so inefficient at its job of grabbing carbon dioxide, plants have had to evolve a brute force workaround. They simply manufacture massive quantities of this enzyme to compensate for its sluggishness.
Which leads to an amazing trivia fact. Because every photosynthetic organism needs so much of it just to keep the system running, Rubisco is widely cited as the most abundant protein on Earth.
Yeah, the sheer volume of Rubisco on the planet is staggering. It represents a massive investment of the plant's resources, all just to keep that Calvin cycle turning at a fast enough rate to sustain life.
Okay, so the plant has this delicate machinery, these slow, mistake-prone enzymes, and it's all powered by light. Here's where it gets really interesting. Does just any light work? Like, if I shine a random desk lamp on a plant, is this machinery picky about the light it uses?
Oh, definitely. Light quality matters just as much as light quantity.
Uh-huh.
And the way we figured this out is honestly one of the most elegant experiments in biology.
You have to picture this. It's 1882. A German botanist named Theodor Engelmann wants to know which colors of light plants like best.
Right.
He doesn't have modern spectrophotometers or lasers, so he takes a single long strand of algae and puts it under a microscope. Then he uses a glass prism to split a beam of sunlight into a rainbow spectrum, red, orange, yellow, green, blue, violet, and he shines that rainbow directly across the strand of algae.
So different parts of the algae are now bathed in different colors of light simultaneously.
Exactly. But how does he measure which part is photosynthesizing the most? He introduces oxygen-seeking bacteria into the water.
Such a clever trick.
Right. These are tiny organisms that will literally swim toward wherever the highest concentration of oxygen is being produced. And when you look through the microscope, the bacteria weren't evenly spread out. They swarmed the algae in two very specific places, the areas hit by blue light, around 430 nanometers, and the areas hit by red light, around 660 nanometers.
Long before modern technology, Engelmann visibly proved exactly which wavelengths drive photosynthesis most efficiently. Chlorophyll A and B, which are the primary pigments in plants, are chemically tuned to absorb blue and red light very strongly. But they do not absorb green light well at all. Instead, they reject it and reflect it away.
Which is exactly why plants look green to our eyes. We are literally seeing the light the plant is rejecting.
Yeah. And if we connect this to the bigger picture, it shows how finely tuned this molecular machinery is to the specific spectrum of our sun. It's optimized for the specific photons that actually make it through our atmosphere.
Once a plant actually makes its glucose using that red and blue light, we kind of have to talk about how they and we actually use it. If you are taking a nursing exam, understanding the mirror image relationship between the plant's chloroplast and the human mitochondrion is, well, it's critical.
Absolutely. Remember that proton gradient we talked about in the thylakoid? The hydroelectric dam where protons flow through ATP synthase to make ATP?
Yeah.
That mechanism, chemiosmosis, is the exact same fundamental principle used in human cellular respiration inside your mitochondria.
It's the same biological technology. But hold on, I'm confused. If plants make oxygen and animals make carbon dioxide, don't we just cancel each other out in a perfect loop?
That is a massive misconception that routinely trips students up. Animals do cellular respiration, yes. But plants perform cellular respiration too. This is vital to understand.
Wait, really?
Yes. Plants respire around the clock, day and night, in every single living cell. They have mitochondria just like we do. They take the glucose they made, combine it with oxygen, and break it down to make ATP for themselves, releasing carbon dioxide and water in the process.
Wait, so plants are consuming oxygen and releasing carbon dioxide right now?
Yes. They only photosynthesize in the light, but they respire continuously. The only reason a plant is a net producer of oxygen is because during the daytime, their rate of photosynthesis is so high that it wildly outpaces their continuous respiration.
Oh, wow.
But at night, when the sun goes down, photosynthesis stops entirely. At that point, the plant is only respiring, meaning it is actually taking in oxygen and releasing carbon dioxide, just like you and me.
So if you're taking notes, underline this: photosynthesis and cellular respiration are chemically reversed processes. Cellular respiration takes glucose and oxygen to make CO2, water, and energy. Photosynthesis takes CO2, water, and light energy to make glucose and oxygen. And plants do both.
Exactly. Understanding that interplay is foundational. Once you grasp how a plant cell handles ATP, electron transport chains, and oxygen, you essentially already understand the mechanics of human cellular metabolism and oxygen transport. It's the exact same machinery running in reverse.
This microscopic chemistry isn't just about passing a test, though. I mean, it has literally shaped the entire planet. Let's look at the global stakes here. Starting about 2.4 billion years ago with something called the Great Oxidation Event.
Oh, this is fascinating. The very first organisms to figure out this water-splitting trick were cyanobacteria. Over millions of years, they produced so much oxygen that it fundamentally altered Earth's atmosphere.
Which was great for us, eventually. But at the time, oxygen was highly toxic to the anaerobic life that dominated the planet. Photosynthesis is arguably responsible for the largest mass extinction event in Earth's history.
Yeah, it basically poisoned the existing biosphere to make way for a new one. And this machinery is still shaping history. Remember our slow, inefficient factory worker, the enzyme Rubisco, the one that grabs the blue oxygen boxes by mistake?
Hard to forget.
Well, Rubisco has a fatal flaw. When the weather gets really hot and dry, plants naturally close their pores to save water. That means oxygen can't escape and builds up inside the leaf. Rubisco gets completely overwhelmed by the oxygen and starts grabbing it instead of CO2.
Oh, that's not good.
No, it creates that wasteful, energy-draining side reaction we mentioned called photorespiration. The plant starts accidentally burning through its own resources without making any sugar.
That sounds like a death sentence for a plant in a hot, dry climate.
It would be, but evolution found a hack. About 3% of plant species, including major crops like corn, sugarcane, and crabgrass, evolved a variant called C4 photosynthesis. They basically created a specialized, physically separate compartment just for carbon fixation.
So they move Rubisco into a VIP room.
That's a great way to look at it, yeah. They have regular cells near the surface that quickly capture CO2 and pump it deep into these specialized interior cells where Rubisco is sequestered. By flooding that VIP room with pure carbon dioxide, they keep Rubisco entirely away from the oxygen, preventing those costly mistakes.
That's brilliant.
It really is. This spatial separation allows C4 plants to thrive and photosynthesize efficiently in extreme heat, where regular plants, like wheat or rice, would lose massive amounts of efficiency.
And understanding that spatial separation isn't just academic. It leads directly to the Green Revolution in the 1950s and 60s. Scientists like Norman Borlaug intensely studied how plants allocate their photosynthetic output.
Exactly. They realized that if you can breed plants to maximize their photosynthetic efficiency, you can change the world.
Right.
By breeding high-yield dwarf wheat varieties that captured energy into growing edible grain rather than uselessly tall stems, they dramatically increased food production. I mean, they averted famine for hundreds of millions of people in places like Mexico, India, and Pakistan.
That is the power of understanding this process. It's not just a textbook diagram, it's the bedrock of global food security.
It raises an important question for how we view our relationship with nature, too. We depend on this process entirely, yet we're constantly trying to improve upon it to ensure our own survival.
So what does this all mean for you? Let's rapid fire the top takeaways for test day. First, the oxygen released in photosynthesis comes from splitting water, not from carbon dioxide.
Right.
Second, it's a two-stage process, light-dependent reactions in the thylakoid making ATP and NADPH, and the Calvin cycle in the stroma using those batteries to fix carbon into sugar.
Spot on.
Third, plants respire continuously. Photosynthesis and cellular respiration are reverse processes happening in the same plant. And fourth, Rubisco is the most abundant protein on Earth because it is so notoriously slow and inefficient.
Well summarized. And I'll leave you with one final, deeply humbling thought from our sources today. Despite being the foundation of almost all life on Earth, the driver of the global food supply and the original source of all fossil fuels, natural photosynthesis is shockingly inefficient.
Oh, really?
Yeah. In most crop plants, it only converts about 1% to 2% of incoming solar energy into stored chemical energy.
Wow, 1 to 2%, that's it.
That's it. Which raises an incredible question for the future. Right now, there is an ongoing high-stakes research race for artificial photosynthesis. Scientists and engineers are trying to mimic this exact light-to-chemical energy process to produce clean, synthetic hydrogen fuel.
Oh, wow.
Yeah, imagine what it could mean for the global energy crisis if human engineering can somehow outsmart billions of years of evolution and beat that 1 to 2% efficiency limit.
That is wild to think about. We started by throwing out the simple flashcard, and we've ended up at the frontier of clean energy. Well, thank you for joining us on this deep dive. Whether you're staring down the TEAS exam or just marveling at the nearest houseplant, remember that the grade school definition is never the whole story. Keep questioning the world around you. We'll see you next time.